Some people like this. Maybe I've got a bigger mouth. Some people like this volume louder than I like. All right, so it's Friday. Exam a week from today. That didn't get a yay, did it? And as I said last time, I will not be here on Friday next week, so Dr. Merrill will be here giving the exam, and he knows how I like to do exams, so give him your attention and seat yourselves appropriate, as I've discussed before. As I said, I will do a review session in class on Wednesday, and material will stop with wherever I stop on Monday, which will likely be the end of this section. Okay? So material will stop on Monday, wherever I end up stopping there. Okay? We've got a fair amount to cover, but actually it's quite doable, and I think we actually may slow down a little bit. I know going pretty fast for some of you, and I apologize for that. A lot of material to cover this term. Well, last time when I finished, I was, in fact, talking about the removal of Okazaki fragments by DNA Polymerase I of E. coli. And so let's go back and think about that. So we're at the replication fork, all right? And this is one representation of the replication fork. I like it because it's simple. And in reality, the replication fork is actually quite a bit bigger, or quite a bit more complicated. This begins to show some of the complexity of that replication fork, and even this doesn't do a very good job because it still shows two separate polymerases when in fact it's one polymerase with two heads that are doing this. This figure illustrates something called the trombone model of DNA replication. And the trombone model arises primarily because of the fact that leading strand synthesis proceeds faster than lagging strand synthesis. So what happens is the leading strand synthesis gets zipping along here, and the lagging strand synthesis, remember, it has to make a primer, it has to elongate the primer, it has to detach, and it has to keep remaking that primer and making new strand as it goes along. So the lagging strand synthesis can't continue as fast as the leading strand synthesis does. So because of that, rather than stop everything, the cell sort of loops out a part of the lagging strand as the leading strand is moving along. Now, the leading strand won't continue forever. It will eventually slow down. But for at least a portion of the time, the leading strand is moving fairly fast, and the lagging strand is being looped out in this sliding trombone model that's there. If you look at the videos that I posted links for online, I think you'll see this trombone model shown reasonably well. Again, they're not perfectly accurate because at least one of them shows two different polymerases, and again, that's not exactly the right model, but you get the idea. And that reminds me also, somebody sent me an email after class last time saying that someone told them in, I think, a micro class, I'm not sure what the story was, and I've heard this before too, that in the class they were taught that the leading strand synthesis was Pol III, the lagging strand synthesis was Pol I. That's not correct. Okay? That is definitely not correct. Pol I participates in the replication of the lagging strand, but the Pol III is still doing it. In fact, you can see it right here. That's Pol III that's replicating the lagging strand. So Pol I comes in and, as I noted, displaces the primers. I'm going to explain that process to you in a little bit more detail in the lecture today. Okay? So, but to say that one is Pol I and the other is Pol III is 100% wrong. Okay? So forget that if they told you that. All right. Well, let's think then about Pol I, because Pol I is pretty critical. Pol I has the ability to remove these primers, and that's single-stranded binding protein there. So there's a primer right there. We've got to remove this primer, okay? And only Pol I can do it. Pol III cannot remove the primer. All right. So if we think about what happens when we have Okazaki fragments being made, okay? So here are a series of Okazaki fragments, and this is again representing what's happening. In this case, the leading strand is going from right to left, and the lagging strand is going from left to right. And the head, that is the very leftmost portion of each of these little blue fragments, has a primer. That primer, of course, is RNA, and that RNA must be removed. So To remove that, what happens is DNA polymerase I comes in and starts at the 5' end of that fragment. So this is the 5' end of the fragment right here, 3', 5', 3', 5', 3'. Okay? So Pol 1 will come in and attach itself to the 5' end of the Okazaki fragment. And it uses an enzymatic activity that it has to remove that primer. Now I need to describe that activity to you a little bit. So it turns out that DNA polymerase I has not one, not two, but actually 3 different catalytic activities. 3 different catalytic activities. One of them, well, it's got a DNA polymerase activity. It makes DNA. All right? The second one I want to talk about here is the catalytic activity that removes the primer. Okay? So the catalytic activity has a name, and it's called 5' to 3' exonuclease. EXONUCLEASE. All right? Well, what does that mean? Well, let's imagine that I am an RNA polymerase, a DNA polymerase I that has attached itself to that 5' end, and it's going to go sliding from left to right. It's going to start moving 5' to 3'. The name exonuclease tells us something about what the activity does. Exo means outside of. Okay? So this DNA polymerase I is starting on the outside of, that is the end of the fragment. And the second part of the name nuclease tells us that it chews up a nucleic acid. So what it does is it starts at the 5' end and it moves inwards, destroying that RNA. Okay? DNA polymerase III does not have a 5' to 3' exonuclease. Right? Well, if we look at this, what we can see, if we started, let's say instead of starting here, let's say I started here. Imagine that I had a DNA polymerase that attached itself here and said, oh, look, I can replicate from this 3' end because this is a primer. Right? This is DNA here. And it's already primed because it's already been made, so all it has to do is attach itself to the 3' end of this fragment. If it attaches itself here and starts putting down nucleotides, it's going to come up here and all of a sudden it's going to hit its head on this 5' RNA, end of the RNA of the next fragment. And the exonuclease says, no problem, I'll start chewing it up for you. So at one end, the polymerase is chewing up the RNA. At the other end, it's making DNA. It's really cool. All right? So it's a Pac-Man at one end, and it's a polymerase at the other end. Well, the result of that is that eventually the RNA gets all gobbled up, gets replaced by DNA, and the only thing that's left is a slight gap between there. And as I said in the lecture on Wednesday, the gap gets joined together by DNA ligase. So the upshot of this is you have removed the RNA, and you've now made an intact DNA fragment as a consequence of that. Now, this basic process that I've just described to you works in human beings as well as in bacteria and every living system on Earth. Okay? The enzymes have different names. We're not going to worry about the names. We're going to, for the most part, focus on the names in the E. coli. Okay? Well, so that's what DNA polymerase I does. DNA polymerase I, you remember, is very progressive, meaning that it doesn't stay on the DNA for very long. It falls off. So once it's gone here a little ways, it gets into here, it falls off. Will it chew up DNA in a 5' to 3' direction? The answer is yes, it will. So it doesn't have to stop exactly where the RNA-DNA junction is. It may go a little ways into the DNA, but it's going to replace it on its backside with more DNA, so it's not a problem. Okay. So that's a unique activity of the DNA polymerase I of E. coli. That unique activity cannot be replaced. If that is destroyed, the E. coli cell will die. Yes, question, Connie? I haven't said it yet. Okay, so the questions back here were, what was the third activity? So I'm getting ready to the big climax to talk about the third activity here in a second. Okay? All right. Now, the third activity that we find in DNA polymerase I, we also find in DNA polymerase III. So this activity I'm getting ready to describe to you is not unique to DNA polymerase I. It's found in DNA polymerase III, it's found in DNA polymerase I, and in fact, it's found in most DNA polymerases that exist out there. Not all of them, but most of them. Okay? Most DNA polymerases have the activity I'm describing to you. Well, let's imagine, let's go back to my better little replication fork here. Actually, that's not a better one. Maybe I'll go back here. All right. So let's say that I'm going down the leading strand. Okay? I'm a DNA polymerase, and I'm moving along here. In this case, if I'm on the leading strand, I'm definitely a DNA polymerase III because we don't see lagging strand on the. We don't see DNA polymerase I on the leading strand. DNA polymerase I is going along, and it sees an A, and it puts in a T, and it sees a G, and it puts in a C, and it sees a C, and it puts in a G, and it sees an A, and it puts in a T, and it just keeps going along and along. Remember, it's going along, doing this at 1,000 bases a second. Okay? 1,000 bases a second. Well, nothing is perfect, and DNA polymerase is pretty close to perfect, but it's not perfect. It goes along, and it sees an A, and it makes a mistake, and it puts in a C. What happens? Okay? Well, DNA polymerases make mistakes, and so it's going along, everything's fine and dandy until all of a sudden it's got this Ca base pair. Well, as you might imagine, a Ca base pair actually doesn't form stable hydrogen bonds. And remember how I said that the dimensions are set up perfectly for an At base pair and a Gc base pair? They don't vary in dimensions. If you try to put an A with a C, it bulges. It bulges. Okay? Well, the DNA polymerase recognizes that bulge. It recognizes, uh-oh, I think I might have made a mistake. And so it stops. It stops right there. Okay? And what it does is kind of cool. The DNA polymerase III and the DNA polymerase I each have a 3' to 5' exonuclease activity. What's the difference? 5' to 3' goes one direction. 3' to 5' goes the other direction. So let's think about this. The polymerase is moving 5 to 3, 5 to 3, 5 to 3. Here's an AC. It says, oops, and it backs up. When it backs up, it's going 3 to 5. The 3' to 5' exonuclease activity is kicked in as soon as it recognizes it's made a mistake, and it starts chewing out bases. It's chewing out, it's correcting the mistake that it made. Really cool stuff. That activity is also called proofreading. Okay? Proofreading is the same as 3' to 5' exonuclease activity. All right? They're the same thing. So this guy backs up and chews it and removes the mispaired base. Now, proofreading is an activity that is present, as I said, in most DNA polymerases. There are some DNA polymerases, however, that do not have proofreading. When we compare the error rates that they have compared to the error rates of DNA polymerases that have proofreading, no surprise, we discover that ones without proofreading make many more errors. A really good example of an enzyme that does not do proofreading is the enzyme that makes DNA for HIV. HIV is its DNA polymerase does not have a proofreading ability. It makes errors at probably about 1,000-fold greater frequency than does a DNA polymerase with proofreading. Okay? Once it makes a mistake, oops, sorry, I guess I'll just keep going along. Well, it turns out for a virus like HIV, that actually works because mutation in general allows for variance. The more variance that you have, the more likely you are to succeed if something is trying to stop you. If one of those things trying to stop you is an anti-HIV drug, you can evolve resistance around it by evolving changes in the proteins of the virus. It's one of the reasons, and there's many, but it's one of the reasons that HIV is very difficult to treat because it will usually mutate its way around a treatment that kills it. Because it's got this very error-prone DNA polymerase. A very error-prone DNA polymerase doesn't work well for an organism. Why? Well, you're much more likely to activate an oncogene, cause a cancer, kill the organism. For a virus that's out there, it doesn't have to worry about that sort of thing. Okay? Very big difference between the two. Okay, so as I said, DNA polymerase III has that activity. DNA polymerase I has that activity as well. At the end of the lecture last time, somebody said, well, you didn't say anything about DNA polymerase II. Okay? You jumped from 1 to 3. What happened to 2? Well, 2 turns out to be a relatively minor polymerase in the overall scheme in E. coli. It's mostly involved in repair and repairing damage. And I really won't address any considerations of it here in this class, and it's really not a significant polymerase in the cell at all. Okay, I will slow down and stop there and take any questions that you might have with respect to that. Yes, sir?
Yeah, so when DNA is proofreading itself,
Uh-huh.
and it backs up. When it
The DNA
backs-
polymerase is proofreading.
Yeah.
Yes, the DNA can't proofread.
Right. When it backs up, does it just delete everything as it's backing up?
His question is, you know, when it backs up, when the DNA polymerase is proofreading and it backs up, how far does it go, basically?
Yeah.
And the answer is it'll actually go back a few bases.
And it just gets rid of everything.
For a few bases, you know, 5, 10 bases. So, and that means it probably removing some perfectly paired bases, but that's fine because now the polymerase comes back in and just fills them back in. Yeah. Yes, Jody.
So it backs up over the error, chews out a couple extra bases, turns around and starts going forwards again, but
Yep.
then hits the DNA that was already there. Does it fall off at that point until a ligase?
Not on lead, no, well, in the case of DNA polymerase III, it's on leading strand, so it's not hitting anything. In the case of lagging strand, when it hits something else that's there, then of course, yes, that's always going to be a cause for it to fall off. Right? Yes, Jared.
If I remember correctly, whenever HIV infects, it inserts some of its DNA into the host, and then would that DNA code for the polymerase?
Yeah, so when HIV infects the host, it puts its DNA in the host, and that includes the coding for the polymerase. The HIV polymerase is a different kind of a DNA polymerase than we've talked about, and since you've asked the question, I'll just very briefly mention what it is. It's an enzyme known as a reverse transcriptase. Okay? This is the DNA polymerase found in HIV. And reverse transcriptase derives its name from the fact that regular DNA polymerase has to have DNA that it copies. A reverse transcriptase will copy RNA. That is, it'll use an RNA template and make DNA from it. A regular DNA polymerase will not do that. Now, I think the upshot of your question is, well, if this polymerase is present in the cell, is it causing problems for the cell? Is that the upshot? And the answer is it may be to a small extent, but the bigger problem of the integration into the cell is that when the RNA is transcribed, it transcribes the whole genome. Okay? And that whole genome now encodes all the proteins for packaging and so forth. So once that genome has been transcribed in the cell is pretty much hosed. So it doesn't, you know, exist for long enough to really have much of a reverse transcriptase as an issue. Okay? Yes, Connie.
When you say that DNA polymerase can do a base pair like 1,000 per second,
Yeah.
does that include proofreading?
So yeah, it's pretty astonishing, isn't it? Her question was, you're telling me this goes 1,000 a second, and that includes proofreading? And the answer is yes, it does. Yeah. I mean, it's pretty awesome. It's pretty darn awesome. And it's not perfect. So even proofreading, you know, you've turned in papers to professors, right? You'll read it 100 times. I'm sure it's perfect, and you get it back and you go, oh man, I can't believe I said so-and-so. Proofreading itself is not perfect, but it improves the accuracy of the DNA polymerase at least 100-fold, maybe 1,000-fold. Yes, sir? Does anything inhibit the proofreading capacity in terms of drugs or something?
Yeah.
Not as far as I know of, no. Okay, yeah, one more.
Is primase part of the whole polymerase complex?
Yeah, very good question. Is primase part of the polymerase complex? In fact, it is. So if we look at the replication fork that I showed here, primase is actually a part of this overall complex, yes. Because if it's not, then you've got to wait for it to come in, and you know, you got this delay, et cetera, et cetera. So the E. coli cells are very efficient. They've got everything there that's needed to do this. There are suggestions that in addition to having all the proteins necessary to make the DNA, and this is kind of a cool thing, that there are proteins, that is enzymes here, that are making nucleotides right at this fork and shooting them at the fork. Why would that be important? Okay. Well, there's a very odd thing. It's debated a little bit, but there's a very odd thing that suggests that the rate of diffusion of nucleotides to the fork, if we assume that we have an equal concentration of nucleotides throughout the cytoplasm, that the rate of diffusion is not fast enough to get them there at 1,000 a second. So then you have to account for, well, how in the world, if they can't diffuse there fast enough, does that happen? There's some suggestions that some of the machinery actually starts synthesizing nucleotides in that direction so they have an artificially high concentration at the replication fork. Kind of cool stuff.
So since those trinucleotides are sources of energy themselves, what provides the energy to synthesize new ones? An excess one converted to another?
I'm not sure I understand your question.
How would you synthesize new ones there? What's the driving force?
What would the driving force be?
Yeah.
The enzymes. I'm not sure I understand.
For the creation of the nucleotides, because they're an activated intermediate that actually goes into those, correct?
So the nucleotides are made from basic building blocks, yeah. So your question is how the basic building blocks get there?
Let me roll it over. I'm sure.
Okay, that's fine. We'll talk later. All right. Let's see. Let's move on. So that's basically what's up with DNA replication in terms of the proteins. There's one protein I haven't said much about, and I want to say a little bit about because it's really interesting also. I think many of these proteins have really cool functions. And this is the enzyme I called topoisomerase. Now I want you to remember that topoisomerase is a class of enzymes, and there's a specific one that's involved in E. coli called DNA gyrase. DNA gyrase. Well, in order for me to make some sense of this for you, I have to tell you a little bit about what are called topoisomers. Okay? So, question? Okay. If we look at DNA, let's say I take some DNA from a cell, and I put it under an electron microscope, which is what's happened here, and I look at it. These are both circular DNAs, and as I said, E. coli has circular DNAs. I discover something interesting. One here looks, on the upper left, looks all kinky and all tight, and the one on the right looks, oh, it's really great big. Well, in fact, these two DNAs have exactly the same number of base pairs in them. Okay? They have the same number of base pairs in them. The one up here on the upper left has been all kinked up. Well, again, you play with rubber bands, you know, if you start twisting and twisting and twisting, you'll see the rubber band, what it's going to do is it all coils up. And that's called supercoiling. That's what's happened to this guy here. Supercoiling plays several functions in cells, one of which is making a DNA smaller. There are other functions that supercoiling has. But supercoiling arises because we have a closed circle. And if we take a strand and we make a little break and we untwist, 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 and we twist it back, what have we done? Well, we've just changed the number of times that the DNA wraps around itself. And since I said that 10.5 base pairs per turn is a magic number, I've just changed the number of turns. What's going to happen to that ratio? It's not going to be 10.5 anymore. That molecule is going to be under stress, and that stress is relieved by this. It's relieved by kinking up. The kinking up happens as a way of relieving that stress. What the kinking up is trying to do is artificially, I shouldn't say artificially, but re-put turns back into it so that it is 10.5 base pairs per turn. And by putting turns back into itself, it kinks up in the process. Now, I've got a better figure to show you that, so you don't have to try to envision it so much. Let's think about this, okay? So there's three parameters we need to think about with DNA. It's called linking number, twisting, and writhing. Okay? Linking, twisting, and writhing. For our purposes, twisting and turns are going to be the same thing. Twisting and turns are going to be the same thing. Okay. There's a relationship between the linking number, the twisting number, and the writhing number. And it's very simple. Linking equals twist plus writhe. L equals T plus W. Now I'm going to tell you what linking is in a minute. But turns are basically the number of twists that we have with this guy. Let's imagine I have a linear DNA. It's got 25 turns in it. And for our purposes of keeping this simple, I'm going to say this is at 10 base pairs per turn. Instead of 10.5 to be relaxed, we're going to say 10 to make things simple. So this guy's 250 base pairs long. It's got 10 base pairs per turn. It's relaxed. It's like you are watching the tube, right? You're sitting there. If I take that DNA and lay it out on the table, It's just going to relax, and it's going to be at 10 base pairs per turn. If I take that DNA and I join the ends, I join this end to this end, I'm not going to change anything. It's going to remain relaxed, and it's going to look like this. It's still got 25 turns, twists, same thing. It has 0 writhes, and it has 25 linking number. Now, what does that mean? Okay. Well, in order to understand linking number and writhes, we have to understand what happens if we change things. Okay. Let's say I take that same DNA. It had 25 turns. It's got the same number of bases, but now I unravel it a couple times. So now instead of having 25 turns or 25 twists, it now has 23. I have the same number of base pairs, So when I put this guy together into a circle, I've got 23 base pairs, but I still have 23 turns, but I have 250 base pairs. This guy is going to be under strain because it's no longer 10 base pairs per turn. Okay? Well, it's a closed circle. It can't relax all by itself. If I have it laid out here linear, it'll just kind of go, and it'll kink back up. It'll put a couple more turns in itself, and it's fine. Well, if it tries to put a couple turns in itself, let's say this strand decides, okay, I'm going to wrap around there once, I'm going to wrap around there again, it can do it. But it does it at the cost, okay, of what's known as the writhes. Right? Look at this. All right? The twists are 23. The linking number is 23. All right? Now this thing is going to say, well, I really want to have 25. Let's get 25. So it wraps itself around a couple times, but that changes the conformation of the molecule. The linking number does not change because, again, I haven't opened this guy up. In order to make this equation be equal, the linking number equals the twisting plus writhes, I had to put in what's called 2 negative writhes. This is supercoiling. When the writhe is anything but 0, the molecule will be supercoiled. Yes? It is back to 10 base pairs per turn, exactly. By putting these 2 turns in there, it's happy. But the molecule overall has a distinctly different shape. Well, where does the linking number fit in? It turns out the linking number is the number of times that the strand crosses over itself, just like the number of turns, right? Plus the number of times the double helix crosses itself. We see that it crosses itself twice. In this case, the crossings are negative. It could also be positive, right? What if I had put in 2 more and made 27 in the twists instead of 25? Well, it's going to uncoil a couple times. And when I uncoil a couple times, I've got to make this be a positive 2. You're not going to have to look at a structure and tell me, is this negative writhe or is this positive writhe? All right? But I think that you should know that the linking number is equal to the twists plus the writhes. You'll always be able to figure out what the writhe is. You should always know that if we have supercoiling, the writhe is something other than 0. Now, I know that is a little complicated, and I know that that is a little hard to get around your head, so I'll stop there and take questions for that. Connie? The twisting number is just simply the number of times that the twists go around and around and around and around.
Okay. Looking at that picture, I mean, like the DNA helix crosses itself.
Yes. That's the writhe. So the crossing, anytime the double helix crosses itself, we get a writhe. Right. Okay? If we look back, now let's think about this. We go back to that picture that I showed you to start this process. Look at this guy. Do you see any writhes there? Do you see the thing crossing itself at all? You don't. Do you see writhes up here? Yes, you do. The helix, in fact, it's a little hard to see how many times it's writhing, but you can see that that double helix is crossing itself many times. This guy is very supercoiled. Supercoiling can be positive or negative, and they both have the same effect. They both will cause the molecule to kink up. Jared?
I'm not sure this is necessarily a fantastic question. It seems like it would affect gene expression depending on how.
Yeah. So his question is, will this affect gene expression? The answer is absolutely. Absolutely. Some genes get turned on by this. Some genes get turned off by this. Yep.
So is this integrated as part of like chromatin expansion and contraction?
Okay, so his question is this somehow related to chromatin? And in fact, this is a consideration in chromatin structure. Then you say, well, DNA in eukaryotic cells is linear. Why don't the ends just ravel or unravel like that thing I laid out on the thing? They're so long that you get local areas of tension that don't get relieved in that way. So yes, it's a factor for chromatin, indeed. Yes, sir.
Is the ultimate reason for this thermodynamics? Is it more energetically favorable to be twisted up like that?
Yeah, so his question is this rooted in thermodynamics? And the answer is absolutely yes. Absolutely. You want to see the thermodynamics?
Yeah.
We're not going to do that. Okay. Yeah.
Right before it supercoils, does the number of links or the number of twists remain the same?
Okay. So let's look at what happened. I'll show you the answer to the question, okay? So here's what we had. We had a relaxed circle, right? We took the relaxed circle and we untwisted it a couple of times. And when we put that back together, we had this, all right? Notice that in this strained state, these two guys are equal. Notice the writhe is 0. The double helix is not crossing itself yet. But this is not stable. It's not going to sit like this for very long. It tries to reestablish its balance, and when it reestablishes its balance, okay, what we see is that the twist goes to what made it stable in the first place. The linking number does not change. That means that the writhe had to change. Okay? So a guiding principle here is if the DNA is at something other than 10, or actually 10.5, but it's something other than 10 base pairs per turn, it's going to have strain on it. And it's going to do something to relieve that strain, and the something it's going to do to relieve that strain is going to be to try to get back to 10 base pairs per turn. In doing so, it's going to have to writhe in order to make that happen. Other questions about that? Okay, well, why do I tell you all this? The reason I tell you all this is what topoisomerases are doing, they're affecting this twisting that's there. Okay? They're affecting that twisting that's there. Let's think about what's happening at that replication fork. Remember, we've got a helicase ripping things apart at 6,000 RPM. The strands are coming apart, and you said that ahead of where that helicase was, if we don't relieve that strain, that the DNA is going to turn into a knot. So I said ahead of that strain, we've got, think about this strain now in a different way, right? We've changed base pairs per turn, right? We start pulling. The other end doesn't give. We're going to have more base pairs per turn. It's going to cause some supercoiling. We're going to see some kinking. And you've seen examples of kinking. It can get really ugly. The topoisomerase ahead of that is known as DNA gyrase. And DNA gyrase, in order to alleviate that strain, has got to basically open up the DNA, okay, and let it unwind at the other end. So what you're putting in as strain at this end has got to be relieved at the other end. So a topoisomerase, okay, is changing the topoisomeric configuration, that is the way that the DNA lays, is changing that twisting, writhing phenomenon. It's changing how the strands are wound around each other. There are two types of topoisomerases in general that we will consider. All right? One type is called type 1. All right? The other type is called type 2. DNA gyrase turns out to be a type 2, but it's a little harder to understand. So I'm going to start with type 1. Okay? Type 1 topoisomerase. Let's imagine I'm back here with this guy right here. All right? This guy is all kinked up. All right? It's got 10.5 base pairs per turn, but that writhing has really made the overall molecule be in a very different configuration than this guy. How did we go from this guy to this guy? It turns out there's a very simple way to do it. All right? Imagine, if you will, that I'm an enzyme and I break one bond between two nucleotides on one strand. All this energy that's stored up in here, what do you suppose is going to happen? Well, so I've got double helix, all right? One strand, it's a little hard to do. Okay, let's see. How am I going to do that? I've got a double helix, okay? And they're sitting here, as we see, and an enzyme comes along and it makes a break in this one strand on top, but not the strand on the bottom. Everybody understand that? This top strand, I can have half of the thing, actually is free to do this around the bottom strand. Right? It's free to do it like crazy. There's nothing to stop it from doing that. And look at all the energy that's stored up in this kinky molecule. What's going to happen when I cut one strand? Well, it's going to be like a spring. It's going to go sproing, and it's going to go bzzz, and unwind. It's going to relieve that strain in a different way. Okay? What I've just described to you is how a topoisomerase I works. It cuts one strand to alleviate the strain, and the strand basically uses its own tension to just wrap until finally everybody's set. Connie?
So does that mean it breaks off the hydrogen bonds connecting the two strands together?
No. So her question is, does this break the hydrogen bonds? It does not. Because all that's happening is the other strand is swiveling around its phosphodiester bond. It's a swivel. Right. So the hydrogen bonds are not broken. The base pairs are not broken. The base pairs are still there. Okay? But we have one phosphodiester bond that's holding that thing together. That's how a topo 1 works. A topo 1 will always work by nicking one strand and letting this guy do its thing. Jody?
Do these attack one particular site, or is it just whenever the tension gets high enough?
Yeah, they don't have site specificity. No. So his question was, does it attack a specific site? And the answer is they don't. How does a topo 2 work? Topo 2s are pretty wild. All right? Topo 2s work by cutting both strands. All right? So instead of cutting just the top strand here, a topo 2 cuts both of these strands and lets the same thing happen. It lets go. You like the sound effects? Okay. It lets go. But the problem is, you got to keep the strands together. You can't just let it go flying off in space and unrenting. In the first case, you had one strand that was holding everybody together. Now you've cut both strands, but you still want to hold them together. You want to hold them so that they don't fly apart, because you want to put them back together when they're done. And that's what DNA gyrase does. It's a type 2 topoisomerase. Type 2s cut both strands, but basically hang on to both ends and let them do their thing without flying away. That's logistically hard for me to get my head around. But that's what they do, and that's how they work. Okay? DNA gyrase requires ATP to do it, and it's partly because of the need to try to maintain some control here in keeping those strands together. Yes, sir?
Do these religate the phosphodiester backbone?
Oh, very good question. His question is, we've broken the bonds. Do these enzymes put the bonds back together? That is, the phosphodiester bonds that were broken? The answer is yes. They do. And that's part of the reason why they need the ATP energy as well. Yes, sir?
From a medical perspective, would it be advantageous maybe to upregulate the expression of DNA gyrase?
Okay, so his question is, medically, does this have any significance? I'll change your question a little bit, okay? Medically, does this have any significance? The answer is yes, all right? Let's think about this. Topoisomerase is essential for DNA replication, right? Are there differences between E. coli's topoisomerases and human topoisomerases, or bacterial topoisomerases and human? And that's how you look for any antibiotic, is looking for a difference between the two, and the answer is yes, there are. And you can find and identify drugs that will target bacterial but not human, and they're used in antibiotics. Okay? Ciprofloxacin will do that. All right? Ciprofloxacin will do that. I always like to tell the story. No, actually, I won't tell the story. I'll hold off on that story. Okay. But there are enzymes that are very specific for topoisomerases in E. coli that will make that happen. Okay, there are inhibitors there. There's nalidixic acid. That's more of a general one. And there's ciprofloxacin, which is an antimicrobial. Okay. What do you guys say to a song? I said at the beginning, this one's going to make me a little nervous. I've never sung this song before. You know I don't sing well. And this is a really hard one to sing. I've never sung it in class before, so I really want your help. It's a Beatles song. Okay. Oh, another Beatles song, and it's called I Want to Hold Your Strands. Okay. Oh, yeah, I'll tell you something. It helps to understand. Paul 3 decrees to meet the cell's demands. It's got to hold the strands. It's got to hold the strands. The key, Paul 3, acts most processively. 'Cause see, Paul 3 has beta clamping hands. This is beta's bands to hold on to the strands as it starts replicating a DNA. The unwinding requires alone helicase, helicase, helicase. Primase starts the primer that Paul 1 can erase. Paul 3 takes the primer and starts the DNAs. It starts the DNAs by using RNA. And when a fragment Okazaki displays, there must be joining that requires a ligase, a ligase, a ligase. Thanks to all the factors and all of their ligands, the cell has what matters to replicate the strands. It replicates the strands. It replicates the strands. Oh, that's enough. Okay. That was fun. Thank you. I've never sung that one in class, and with good reason. You heard why. I don't do those high notes. I don't do any of the notes very well, but the high ones are even worse. So, okay. All right. Now, we've got just a little bit of time left, and in that time that's left, I want to say something about how the process starts. And I'll probably finish with that today, okay? So somebody asked me last time, well, does DNA replication just start randomly or what? Okay? Well, it turns out that the overall process always starts at a specific place. It starts at a specific sequence in a DNA called the origin of replication. All right? The overall process starts at something called the origin of replication. E. coli has one of these. We hope it does. There we go. Okay. Now, the process starts with the binding of a protein called DNA. So DNA is a protein that recognizes and binds to some specific sequences in the E. coli genome. All right? In this case, you see 5 of those sequences that are present. And we are looking right now in a region of the overall chromosome called the origin. In the case of E. coli, it's called ORI-C. O-R-I, and then a capital letter C. ORI-C. Okay? You'll notice that next to these binding sites for the DNA, there are regions of DNA that are AT-rich. That turns out to be important. Okay? Now, when DNA binds, it turns out that it causes DNA to coil. It causes it to wrap. What has it done? It's just changed its writhing. That has put some stress on the DNA, and the DNA will act to relieve that stress. And how does it relieve that stress? Well, we've seen winding and unwinding. This guy's going to unwind, and it's going to unwind at the weakest place, the place where the strands can most easily come apart, and that's where the AT base pairs are, because that's where the fewest hydrogen bonds are between the bases. This region right here will open up. The strands, we're not breaking bonds now. I mean, we're not breaking covalent bonds, we're breaking hydrogen bonds. The strands, we see a loop that goes up here, and we see a loop that goes down here, and that big opening is a place for a complex known as DNA BC to bind. So we get DNA A binding, opening of the DNA, and DNA BC binding. Now, DNA BC has something very cool associated with it. It's a helicase. It's a helicase. Okay? Well, what's a helicase going to do when it gets in there? Okay? Here's single-strand binding protein helping to hold it open. There's B, and actually C has left by this point. But DNA BC binds, C leaves, and then we're left with this guy. This guy is a helicase. Well, a helicase is going to take and it's going to start peeling strands, right? And isn't that exactly what we need to do in order to get replication started? You got it. Primase is going to come in. It's going to make an RNA primer, and we've started the process. The whole process happened because DNA A bound to specific sequences next to this AT-rich region. Yes, sir?
Is that just a standard Tata box?
No, it's not a standard Tata box. Tata box is used in transcription. So it's AT-rich region, but it's not a standard Tata box, no. Okay? Yes, Jared?
Is there regulation of this process?
Is there regulation of this process? You betcha. There is regulation of this process. Cells have to coordinate the replication of this, and virtually every cell on Earth has some sort of control over that. In our cells, we have a very intricate set of controls that I'll talk a little bit about in the lecture next time. Okay, I think that's a good place to stop. Let's call it a day, and I'll see you guys on Monday.